The other lesson is that we learned that early therapy is more important than later therapy, so what is lost is not regained.
Another aspect, I think, in the therapeutic arena that may be interesting is other methods of immune intervention and bringing the B-cell depletion into the CNS.
With this cocktail of antigens, one tackles the holy grail of autoimmune diseases, namely re-establishing tolerance.
Hi, I'm Ludwig Kappos. I'm leading the Research Center for Clinical Neuroimmunology and Neuroscience in Basel.
Yeah, my name is Heinz Wiendl. I'm a neurologist, neuroimmunologist based in Münster, where I'm the head of the Department of Neurology. Soon be moving to Freiburg as of October.
And I'm Drummond. You're listening to the ECTRIMS podcast, the official podcast from the European Committee for Treatment and Research in Multiple Sclerosis. Many people regard multiple sclerosis as one of the greatest medical success stories in terms of the advancements made in the treatment and management of the disease over the past couple of decades. However, as researchers continue to make new discoveries that improve our knowledge of the pathogenesis of the disease, this brings with it opportunities for novel therapeutic approaches, which are more targeted and more personalised, to be developed. My guests in this episode, Heinz and Ludwig, have themselves made significant contributions to our understanding of MS over an extended period of time, and today will discuss with me what they think the future holds in terms of therapeutic advances in MS. Heinz, Ludwig, thanks for joining me.
Pleasure.
Yeah, our pleasure.
So, as a starting point, I guess before we move on to what's coming next, I'd love to hear your thoughts around what has been achieved so far in terms of the treatment of MS, and then what do you think are the most promising avenues currently being explored to move us forward?
Well, if I may start, I think nearly all of us agree that life has really changed for people who get the diagnosis of multiple sclerosis, that in the great majority, these people now have the perspective of living a normal or nearly normal life for the next decades. And this is, of course, after a long period when no treatment was available, a major success of both basic science and search for the causes or for better understanding the pathogenesis of the disease, but also of clinical trials that have provided now treatments that can really allow people to have this good perspective.
Yeah, I may add to what Ludwig just said. Completely agree that the perspective, the future of MS patients diagnosed has completely changed. What we've learned are three key messages. First of all, that interference with the immune system matters with regard to, let's say, elements of how we define the disease, either based on inflammatory activity like relapses or in terms of disability progression. So therapy matters. And that was a question in the early days, whether we really can change the fate of the disease. That's one thing. The other lesson is that we learned that early therapy is more important than later therapy. So what is lost is not regained. So we've moved towards earlier, not only diagnosis and diagnostic possibilities, sensitivity and specificity, but we also realized that early treatment is better than later treatment. And the third message is also something that I would like to point out strongly, that more efficacious therapy seems to be statistically superior to less efficacious therapy. And this is part of the blessings we have got over the last one can say decades, that we have now various therapies, more than 20 in some parts of the world, that also differ not only in their modes of action, but also in their potency. So in their efficacy how they halt the disease or how they balance relapse activity, up to a point where some patients end up having statistically one relapse in 10 years or in 20 years. So that's quite significant. And ending this, let's say, intro to what we have achieved is that we have learned a lot by these achievements, and this is probably then leading to the other part. So, what is unmet needs? So, what have we missed in a way? But I think this is the three main messages that I would see as therapeutic key lessons we learned.
Perhaps to add to Heinz's statement, I think we have also become much braver in the application of more intensive and more effective therapies. And this of course, has been facilitated by the increasing body of more or less, but mostly systematic experience with low-key adverse event profile of most of these interventions. And this makes us, although the final proof of the superiority of very early, very high efficacy treatment in a randomized trial is still lacking, made this accepted, and observational studies have also underlined that it is really an avenue that we should follow, even based on the current evidence. So I think that's something that helps us. But as Heinz said, by getting hold of this problem, or managing this problem, we also see where we are not able to do something for people with MS in the progressive phase, or that there are first breakthroughs, but they are much too weak and not effective enough. And that's also where we have to draw our attention and our next efforts, or current and next efforts, to better understand this progression, what is driving this progression, and to characterize the individual patient in a way that allows us to see how much is that for the individual patient or a person with MS and a real problem or not, who is more in need of these additional and hopefully in the future available interventions.
I guess what's clear is while we've been really successful, there is still work to be done. And I think what we see by the number of people who turn up to Congress every year at ECTRIMS, that there is a real commitment to continuing to answer those questions. Shifting the discussion to what is coming next, I'm really interested to know what the two of you see as, I guess, those unmet needs, and what approaches you're particularly interested in seeing as holding promise for the future. Heinz, maybe we'll start with you this time.
Yeah, I have to say that I would make this up as two key work streams. One is the work stream of getting optimal management prognostication of early multiple sclerosis. That means extremely early diagnosis, specific diagnosis, but then also a more individual prognostication. So what we're currently doing is a little bit, it is successful, but it's not very personalized. So from that viewpoint, you would even say, not knowing it better, given the benefit of the doubt, we rather choose the more potent treatments, because that is what is statistically the most likely to give benefit in terms of disability accrual over the years. On the other hand, we're probably over-treating, let's say, 20 to 30%. And we have actually the armamentarium of different types of treatments, but we don't have yet the ways to really personalize the need for treatment. So immunological phenotypes, for example, or things like biomarkers that help us better guide the intensity or the type of the treatment. And I think Ludwig, but also our group, we're trying exactly to get better in this management of early MS. And the first point point is not about new treatments so much. It's to use the treatments and to better apply them individually. The second point is understanding and management of progression. And this is, I think, the clearest unmet need, because we've learned with the therapies that are basically stopping focal inflammation, no more relapses, not very much new lesions, that there is some underlying progression that comes independent of relapse activity or comes as primary problem, like primary progressive MS. And the immunological treatments that we're applying, working mostly outside of the brain, they are obviously doing only partially, if at all, the job. So the promise that I'm seeing is not only understanding it, but also getting treatments that tackle processes better within the CNS. And this is part of the questions that the novel treatments in the arena are trying to address. So how do we measure, but also get a better grip on these elements of progression? And there is a couple of approaches. Of course, there is a lot hope on the BTK inhibitors, Bruton tyrosine kinase inhibitors. We can dwell deeper here with the argument that it tackles B cells, which is a very successful principle already. But in addition, innate parts of the immune system, myeloid cells, microglia cells within the brain. This gives at least a good rationale, going beyond a better and a more reversible B-cell therapy. The other problem is that we don't know until we have the real data in particularly progressive patients whether this rationale is correct or whether it makes such a clinical difference. And we have seen a first disappointment with one of the candidates, evobrutinib, where a lot of discussion has now been kicked off why this has not shown superiority to a, let's say, standard treatment like teriflunomide. So just to make this up, this understanding and tackling progression, I think, is the biggest unmet need probably. And there is some treatments that at least show in their narrative that they could do it, but yet we don't have clinical evidence for it. Not sure what you think about it, Ludwig, because you've done a lot also conceptual work on defining and measuring progression independent of relapse activity.
Yeah, I fully agree with what you said. The challenge is, on one side, to better understand the pathology that most probably mainly in the CNS drives progression, and on the other side, to get a better access to that with our therapeutic interventions. And these possible therapeutic interventions are twofold. One would be to just apply our principles that have helped us to achieve what we achieved in the periphery. So just get the drugs into the CNS and let them do the same thing. And the other is to not only do the same thing, but to adapt to the specific needs or dysregulation or pathology within the CNS. And BTK inhibitors were very attractive to, I would say, all of us, because they promised both. On one side, taking as a small molecule the same principles from the periphery into the CNS, and in addition, adding something, a new way of having an impact on microglia that are, as we understand now, one of the key drivers, at least part of them, of progression. And on the other side, Heinz also mentioned that the disappointment with the first study, which we must admit did not really capitalize on the real possible advantage of BTK inhibitors, because it was full-scale phase 3 program that addressed relapsing MS, where we have already, through interventions in the periphery, a very good result. And it was, of course, disappointing that it was not even better than drugs that are already in use and are seen as medium efficacy. But it was not the right question to answer, and we still hope that some of the drugs that have been are now in development and in phase 3 studies, especially those that address directly progressive MS, that they will make it easier to understand and hopefully also to see a positive effect on progression. So this is still out there. The other aspect that has dampened our optimism to some extent was the issue with rare but still severe hepatotoxicity, which is typical for a small molecule which is not so well targeted as a monoclonal antibody, for example, but interacts with many different cells within the body and therefore has always the potential of creating adverse events, perhaps additional unknown benefits, but also adverse events. And this is another problem that we will need to address with the development of BTK inhibitors. Another aspect, I think, in the therapeutic arena that may be interesting is other methods of immune intervention and bringing the B-cell depletion into the CNS. And this is something that promises to be done with currently available CAR T cells, which have the advantage that they really get into the tissue, also into the CNS, and can therefore exert the same action that monoclonal antibodies, per definition at the moment, are doing in the periphery. So this would be some possible advantage and would help us to understand if this mode of action is sufficient. This would be serendipity at the end of the day, because even B-cell treatment may be somewhat more specific than unspecific immunosuppression like alemtuzumab or perhaps cladribine does, but are still not the specific therapies or immune interventions that we were dreaming of since now more than 30 years, and have been quite efficient in experimental models, but not yet in humans. And at the moment there is one trial out there by the group of in Zurich, which and I think you are also involved in that, Heinz, addressing especially prepared or using especially prepared erythrocytes that are given to the patient in the hope that they will induce a positive counter-regulation of the pathogenic process. So this is at the moment the only intervention that tries to get a more specific hold on specific mechanisms within the immune system.
Yeah, fully agree with everything. Just to deepen what you just said, Ludwig. So with the CAR T cells, it's basically the idea to get a tissue penetrant, more or less complete B-cell depletion. And that is a very elegant thought. It's more or less repurposed from oncology, with the advantage that we really get the answer, is it B cells that we're missing within the tissue that still drive progression? There is, by the way, also a dose increase study running with a already approved B-cell depleting principle, ocrelizumab, based on exactly this thought. We're not getting enough B-cell depleting antibody into the tissue, and if we get more into it, we get more impact on progression. We don't know whether this is true, but this would be an extreme elegant experiment, more or less learning in reverse pathogenesis via a therapeutic intervention. Therefore, it's extremely interesting. The other end of the spectrum, and I very much agree with Ludwig, is to get more specific and selective, because with everything we're doing currently, we are not specifically modifying the process of multiple sclerosis. We're modifying immunological elements of the disease that are relevant to disease evolution. And this is where this tolerance reinduction mechanism comes up. The study is called RED4MS, and it's that Basel. It's a mixture of various peptides that have all been more or less credibly considered to be target antigens of the deviated pathological process. And with this cocktail of antigens, one tackles the holy grail of autoimmune diseases, namely re-establishing tolerance. And this is something we have not really been able to show. First of all, on the other hand, it's also not so simply to show because we would have to know what we are tolerizing against. Means we have to know the target antigen. And this is what makes MS life more difficult than, for example, NMOSD or MORC-associated disease, where we are pretty confident that we've identified the dominant key antigen. That seems different in multiple sclerosis. Many people think that if there is dominant antigens, they are particularly dominating in the early phases of the disease, and then these elements of progression may be not that much depending on an antigen-dominated immune response. And this is what makes this trial so attractive, because it's a standout in terms of the concept. All the others are more or less selective immune modulations that target cells or pathways that are not exclusively relevant to MS, I should say.
I'm interested just to go a little bit more in the tolerance induction, because I think it's a really fascinating approach, and we're, I think, far too early really to be able to answer this question, but I'm going to ask it anyway, just as a point of discussion. One thing that we know that happens is that there's probably bystander activation as part of this process. Do we think that, in terms of tolerizing, it's going to need to be that initial insult, or do you think that we would end up with some tolerogenic, I think the term anti-vaccine has been used at times, considering what it entails, where you would cover all of those different targets? Like, procedurally, how do we think we might approach this?
Well, I think that this has been the aim for many, many years. And what we have seen is that in practice, even if in with some of these interventions you have an indication that some people may benefit, there are others where this kind of immunomodulation or tolerization leads to adverse events. And this has led to stopping some of the trials. One of them I was also participating due to adverse events. And so I think and this leads us to a very important question where Heinz is, of course, very far forward, going very far forward with better characterizing the individual immune response. So what is really driving the process in the immune system? What kind of specific dysregulation exists in groups of patients and perhaps at the end in individual patients. And then we are able to apply not just by serendipity but in an informed way specific interventions that are not only based on insufficient animal models. I think that's a key issue. So better characterizing the immune system, both specifically the cellular part, and this is technically very difficult, very, very complex, and needs a combination of high-end immunology and data science in order to absorb and to work with this high amount of information. I think that's one aspect. And on the other side, and Heinz already mentioned that, it's also our aim, getting more granular in the assessment of morphological consequences of the disease, for example, with advanced imaging. Cristina Granciera in our group is in the forefront of this development. And then also the clinical picture. We are still relying, when we then at the end of the day want to prove a new therapy, we are relying on very crude methods like the EDSS or neurostatus EDSS, which are sophisticated but don't capture really what happens and what is perhaps also important for the life of people with MS. And therefore adding here new aspects, new views, for example, with digital methods that accompany the patient and in a more or less gamified way allow a continuous observation of the state of the disease, of the potential deficits and their development. That together would help us also to be more specific in our approaches, together with more specific approaches, selecting the right patients and the right timing within the long course of the disease. And that's, I think, also something where I guess we really agree with Heinz. Future clinical trials will need not to recruit from the whole population of people with MS, but to recruit according to the specific principle that they are investigating, the specific group of patients. And how to get these specific groups of patients, this must be based on well-documented and well-observed cohorts of people with MS who are documented in centers and in the collaboration of several centers around the world in a way that allows this characterization and then the selection for specific interventions. This makes, I think that's, for me, that's the only way how we can proceed in this phase of the disease, unless we find a vaccination and everything is is behind us. But unless more or less a miracle occurs, and there is no way around that. And I think also pharmaceutical companies or corporate sponsors and public agencies, research agencies realize that, and that it's necessary to invest on that in the long term.
Yeah, this is, I think, a good time point to, it's not really a spoiler, but it's exactly what Ludwig says. We have to acknowledge that the world of MS has, positively spoken, changed a lot. Negatively spoken, it's getting more and more difficult for any new treatment to show really an added value, a benefit. And when we look at how trial populations look like, the conclusion is that how can you show a superiority if with current treatments you have one relapse in 10 years? So is relapse really a endpoint that is relevant to really pursue further? So that means we have to reconsider the classical trial designs. We have to reconsider the heterogeneity of study populations, and all this has led us to initiate an activity which we call the MS Act. And we're trying to bring stakeholders and people together that, the strongest minds in the field, but also stakeholders like the regulators, but also pharma that has the data, to formulate the thoughts how this can now go into the future. Because if we just continue the trial designs that haven't been done for the last 10 or 20 years, we have a high likelihood not to be successful with future trials. And I guess for methodologists, I mean, we're not talking about the specific principles here, but we're talking about the methods to show the value for the MS community in the future. This is exactly a key aim for the community, which is strongly related to the question, so what is causing and what is modulating the disease? And to add on that, very frequently mentioned culprit is EBV. EBV is extremely hot in the topic, even with regard to preventing MS. And there is two things to it. One thing is there is a certain likelihood that it contributes to element of disease propagation beyond disease initiation. I think for disease initiation, it's fair to state it's a necessary but in itself not sufficient condition. But if it is an element of disease propagation, of course, it's worth to look at its modulation. And for only this reason, it's worth to look at it because it's at least a testable hypothesis. I mean, we mentioned that it's difficult to measure the antigens that are driving the disease. For EBV, we can at least measure the response against this antigen, whether it's directly causal or indirectly contributing. And just to again say this is some promise to the field. On the other hand, we have a disappointment seeing that a CAR T-like construct that is directed against EBV expressing B cells, if you want to call it like that, it's a EBV-specific CAR T approach. So that means not all B cells, just those that are presenting and expressing EBV are eliminated and depleted. That did not show a clinical benefit in a phase 2 trial. This is, first of all, a disappointment because it seems not to be so easy. For me, that is not yet the end of the story, but it again is food for a lot of rethinking because it's obviously then not only the EBV expressing B cells that run the show in this progression scenario.
I mean, it's interesting to hear you both talk about clinical trial design as being really important. It was also something that came up in a recent episode that we did around myelin repair and progress being made in that, as that this is something that we need to be putting a lot of attention to, because the last thing that we want is to miss something that could be successful, not because it doesn't work, but because the trials haven't been designed in a way where we can measure success effectively.
Maybe because this is a podcast of ECTRIMS, we should also mention that ECTRIMS has been very active in promoting collaborative approaches to these problems and in specific workshops. And of course, the annual ECTRIMS is the best forum where the results of this work are presented before they, in most cases, before they have been published in peer-reviewed journals. So it's a good opportunity for everyone to keep updated and get the last news on an international basis.
Yeah, and to your specific point on, let's say, maybe non-immune interventions that are directed towards repair, regeneration, protection. Very often it's said they should come ideally in combination, and ideally in combination, in my opinion, to something that is extremely strong treatment. So you would, the ideal trial scenario is you're eliminating more or less what you can eliminate in terms of focal inflammation, and then what is left over is the unmet need, call it progression or call it repair. And that would be, for me, a thought to demonstrate what an additional principle can bring on the table. Because the problem, and then we are back to the trial design, is how can you prove that something that is just remyelinating and not modulating inflammation does do the job. So in the presence of inflammation, it's very hard to show such a effect. And this is in conflict to what regulators expect. They expect a very clean trial design and a superiority design. And that's a bit of a conundrum, in my opinion, for exactly those things like repair or remyelinating therapies. I'm not sure what you think, but I think we're not quite that far with a good. There might not be something like a blueprint of the ideal trial design for such questions, but on the other hand, with the classical endpoints, clinical endpoints, we might simply fail too often, and then the disappointment is too big, and we have to come to a point where also paraclinical markers, surrogates, at least for the earlier phases of the development, are accepted and may be then acceptable also in up to phase 3. Yeah. So that's my key concern, that we're losing time with these trial designs a lot.
Yeah, I fully agree, and it's an urgent need to change the concepts of clinical trials, and this would be another topic for at least one or two podcasts. We won't cover that. We can just express our opinion or belief that this is a very important problem that will be critical for further success, that then also translates not only into more knowledge, but also to better lives for people with MS.
Well, I think you've summed it up perfectly there to say that this is a major topic that we could spend a number of podcast episodes talking about. We've talked about a number of really interesting things on the episode today, and I thank you, Heinz, and you, Ludwig, both for your time today.
Always a pleasure being with Heinz in such a discussion and seeing how much we agree and can help each other to further progress.
Definitely, Ludwig. It was a pleasure.
And thanks everyone for listening. Once again, I'm Brett Drummond, host of this podcast episode. I'm also co-founder of MS Translate, an independent resource centre that aims to simplify the complex wealth of information about MS for the greater MS community. The ECTRIMS podcast is produced and hosted in collaboration with two key partners, MS Translate and the MS Journal, a peer-reviewed international journal that focuses on MS, neuromyelitis optica, and other related autoimmune diseases of the central nervous system. The ECTRIMS podcast is supported by an educational grant from Roche, Alexion AstraZeneca Rare Disease, and other grant providers. Educational grant providers have no input on the podcast series content. You can read more about the topic of this latest podcast episode and more on the ECTRIMS website, ectrims.eu. Do subscribe to the ECTRIMS podcast on your favourite podcast channels and follow ECTRIMS on X, formerly Twitter, LinkedIn, Facebook, and YouTube.